this video covers the IGCSE Biology Objective 7.4.4 on the 2025 CAIE syllabus to describe the locations of digestive enzymes. So let's remind ourselves what the enzymes of the digestive system are. So we have starch that is insoluble and large, being broken down by an enzyme called amylase, which creates small soluble molecules called glucose. We have large insoluble fats, which are going to be broken down by lipase into soluble molecules called fatty acids and glycerol. And we have large insoluble proteins, which will be broken down by proteases into small soluble amino acids. So let's look at the locations of these enzymes through the digestive tract. So in the salivary glands, we have salivary amylase, and we'll see why we are using the term salivary to identify the enzyme as we move on. In the stomach, we have a gastric refers to stomach, a gastric protease, which we will learn the specific name of later. And in the small intestine, we have pancreatic amylase. So there is a second amylase that is released into the small intestine, a pancreatic protease, so another protease that is released into the small intestine, lipase, and another carbohydrate called maltase, which we'll learn about later. So these are the locations of the enzymes in the digestive system: the salivary glands, the stomach, and the small intestine. Now, let's talk about the pH of the enzyme environment for each of these areas. So the salivary glands in the mouth are going to have a pH of 7. So that is the optimum pH for salivary amylase. The stomach has a pH of 3, around 3, due to its acidic environment, and that will be the optimum pH for this gastric protease. And the small intestine is going to have a pH of 8 due to the bile, and we'll learn about that later. And so that is going to be the optimum pH for all of the enzymes that work in the small intestine. So let's talk about this digestive pathway for enzyme action. So in the mouth, salivary amylase begins the digestion of carbohydrates such as starch in a pH of 7. Once the bolus is swallowed and moves down the esophagus into the stomach, the salivary amylase will stop digesting carbohydrates because the stomach has a pH of 3, and so that enzyme will become denatured. At this point, the gastric protease will start the digestion of protein. Once the bolus becomes chyme in the stomach, it will then be pushed along into the small intestine. At this point, the gastric protease will stop digesting the protein due to the pH of the small intestine, which is 8, and so that enzyme will become denatured. Pancreatic juices, therefore, will then start and complete digestion of lipids, carbohydrates, and proteins in its optimum pH of 8.